So cellular respiration, I leave the word cellular off, but respiration is taking glucose in the presence of oxygen and extracting the energy from the bonds in the glucose and turning the glucose into carbon dioxide and water. And again, this is just the reverse of photosynthesis reaction. Of course, the details are very different. The steps in the metabolic pathway are very different. So if I balance this, this would be a 6, 6, 6, 1. Photosynthesis is an anabolic reaction. Respiration is a catabolic reaction. Exergonic, gives off energy. That energy is used to charge up ATP. That's the whole point. It's important to note that plants need to get energy from the glucose they make, not just animals and other organisms, all eukaryotes, basically. So don't just think it's an animal thing, respiration. So this is repetitive but important. Photosynthesis uses energy because it's an anabolic reaction. So it uses energy. The energy comes from the sunlight. Some heats, every time there's a chemical reaction, some heat's released. That's the second law of thermodynamics. We're kind of ignoring that right now. And then, in respiration, which is the reverse of photosynthesis, that's catabolic. We got the sugar being broken down, and that releases energy. Some of that energy goes to ATP, and some is lost as heat because of the second law of thermodynamics. So cellular respiration is basically the reverse reaction of photosynthesis. And to reiterate, anabolic is endergonic, and catabolic is exergonic or exothermic. So this says glucose can carry energy for a couple minutes to 2 hours. But ATP, which is formed by breaking down glucose, only lasts for seconds or minutes. And so ATP is a very short-term energy storage. This really doesn't even store it. It's instant energy, basically. And that's why I call it the energy coin. Instant in the sense of seconds to minutes, and then it breaks down to ADP. So ATP is energy coin. You can't use glucose to run reactions. You gotta use ATP to run reactions that are anabolic, unless they're photosynthesis reactions, and those use the power of the sun, the energy of the sun, not ATP. When we get into the reactions of respiration, we have two other ATP-like molecules, like batteries, that can be charged up and discharged and then charged up again. Do you remember we had NADP in photosynthesis? It's like that, but it's NAD and FAD now in respiration. These are again like batteries. They charge up then they discharge, and then they get charged up again. Okay, so NAD plus is the uncharged form of the battery. This molecule that acts like a battery, and then when it's charged up, it becomes an NADH2. I just write it as NADH. And FAD is the uncharged form. Then FADH2 is the charged up form. So NAD is one of those ATP-like molecules that stores energy for a short time. And which is like a battery, an energized battery and a de-energized battery. So which one is the energized form? The bigger one. So I'll just say this is a charged up battery. NAD is a discharged battery. When I say discharged, that means it's lost its energy. When I say it's charged up, it's storing energy. So when the charged up goes to the discharged form of NAD, of course, energy has to be released. So FAD is, again, one of those special ATP-like molecules that is only found in respiration, some of the steps of respiration. And which one is the energized battery or the charged up battery? The bigger one. And FAD is the uncharged battery. So if that's going to happen that way, energy is again released. Now, if this was backwards, FAD turns into FADH2, energy is stored. So we can do two types of respiration. Aerobic respiration requires the presence of oxygen. It needs oxygen to happen. And then we can do an means without air. We can do anaerobic respiration, which doesn't need oxygen. Okay, so, but anaerobic respiration, that's what yeast does when it ferments, when it gets its ATP. It doesn't use oxygen, and it's very inefficient. You don't make very much energy. You don't get very much energy from the glucose when you do it that way. But it's useful for us when we're sprinting and running out of oxygen, and our muscles need ATP, and so we can do that. We can do short-term bursts of energy without breathing hardly, and make our ATP that way. But it's not very efficient. You can't do long distance running anaerobically. You can do sprinting. So we're going to start talking about aerobic cellular respiration. It happens in 4 stages, and we're going to talk a little bit about each one of these stages. Glycolysis, where glucose is broken down in half and some energy molecules are charged up. PVA, pyruvate oxidation, talk about that coming up. The Krebs cycle, not to be confused with the Calvin cycle. And then oxidative phosphorylation, which is electron transport chains where FADH2 and NADH are cached. They give their energy to ADP to make a bunch of ATP. So each of those stages we're going to talk about. So, first of all, before we talk about the stages, where does cellular respiration occur? Well, it occurs in that powerhouse of the cell, that organelle called the mitochondria. All eukaryotic cells have mitochondria. So let's just review the structure of a mitochondria. We have the outer membrane. It doesn't really show it as two things. Then we have the inner membrane. Inner membrane is highly folded, a lot of surface area. Okay, just like the chloroplast has a lot of surface area, an inner membrane making the granum and the stacks which are stacks of thylakoids. So these things are called the cristae, these little fingers that the inner membrane forms inside the mitochondria. We can see some DNA. Remember, mitochondria and chloroplast is DNA. We can see some ribosomes. Okay, but anyway, this section right there, which they didn't label, is very important. That's called the matrix. That's like the stroma. That space is like the stroma in chloroplast. Okay, so here's a good overview of the 4 stages. We got stage 1 of respiration, aerobic respiration, of course. We got glycolysis, first stage. We got PVA oxidation. We got the citric acid cycle. We got oxidative phosphorylation. Okay, so the stage that charges up the most ATP is which one? Which of these 4 stages charges up the most ATP? Stage 4, look, 34. Per 1 glucose molecule, you get 34 ATPs. Stage 3, you only make 2 ATPs. When I say make, I mean energize. But you make a bunch of NADH2 and FADH2, which will be converted in the 4th stage into ATP. That's why you get so much ATP, because those guys will get cashed into ATP in the 4th stage. So the citric acid cycle, also called the Krebs cycle, charges up a whole bunch of NAD and FAD to make these high energy molecules. So that's a very important cycle right there. But it only makes 2 ATP ones. The second stage doesn't make any ATP or NADH or FADH. The first stage makes 2 ATPs, then 2 NADs. Okay, so those NADHs will get cashed in to make those 34 ATP over there. Also, 2 ATPs are formed in that first stage. Okay, let's not complicate this first stage too much. Okay, so in glycolysis, 6-carbon glucose is broken up into 2 molecules, eventually of a 3-carbon sugar called PVA. So we got the glucose, which is 6 carbons, breaking up into 2 3-carbon PVAs. Okay, so we don't have to look at that too much, but the net result is you charge up 2 ATP molecules and you charge up 2 NAD molecules. So the net result, energy result, is 2 ATPs, like I said, and then 2 NADHs charged up, and 2 pyruvates, which will go on to the next stage. So we see the PVA, the 2 PVAs that were produced here will go into, one at a time, into the second stage of aerobic respiration. Okay, let's summarize the second stage, the PO stage, PVA oxidation. Okay, so basically, remember we have 2 pyruvate molecules with 3 carbons each. I'll just draw one of them. So what happens is this enzyme, which I don't really need you to know, causes one of the carbons to break off of this, leaving a 2-carbon acetyl-CoA, another molecule, because that's what feeds into the Krebs cycle, a 2-carbon sugar, little 2-carbon sugar. So when that breaks off, energy is released, and that energy is used for each pyruvate. It makes charges up another NAD to NADH. So we get energy molecule again that later on will be cashed into ATP on that 4th stage. So here's the carbon dioxide that we breathe out. That's where it comes from. It comes from this step. So the next step is that 2-carbon acetyl-CoA gets fed into the Krebs cycle right here. So here's a 2-carbon acetyl-CoA. Carbon dioxide has been given off. The NADH has been produced in the second stage. So it's feeding into this citric acid cycle, or the Krebs cycle. Now, this is all occurring in the matrix. Very important. It takes place in that soupy stuff inside the mitochondria. You can see it. This is peach. This pink colored region. So glycolysis produced 2 PVAs, which are those 3-carbon sugars, and then they were in the second stage, they were broken up. A carbon dioxide was given off into 2 acetyl coenzyme A's, and you only can put 1 acetyl CoA in at a time into the Krebs cycle. So the cycle has to go around twice in order to use what came from the original glucose molecule. Now, notice a few things here. Carbon dioxide again is produced as the carbons shuffle around in different kinds of molecules going around this cycle. Okay, so let's see. So the 2-carbon hooks onto a 4-carbon, and it makes a 6-carbon citrate or citric acid molecule. And then water's knocked off and the carbon's shifted around. And then water's added. This is kind of roundabout. I'm not going to keep on talking about. The point is carbon dioxide is released, in at two places, just like it was released in the second stage. ATP is formed, it's charged up. NADH is charged up. Some FADH is charged up. A lot of NADH is charged up. Actually, 6 NADHs per 1 glucose molecule. And then 2 ATPs, because it goes around twice, the cycle, for 1 glucose molecule. And then 2 FADHs for 1 glucose molecule. So after 2 spins of the cycle, you have 2 ATPs, 2 FADHs, and 6 NADH molecules, all energy molecules charged up. Now these guys will get cashed into ATP during the final stage, oxidative phosphorylation. Okay, so let's do a little review here. The total products of glycolysis are, okay, you get 2 3-carbon sugars, so that's true. We get, okay, so we don't just get that, we get 2 NADHs, remember, and we also get 2 ATPs. And so this is the right answer. Hey, this was like a really confusing question, but it really isn't. 3-carbon pyruvate is converted into acetyl-CoA. And an extra carbon atom. Well, the extra carbon atom is attaches to oxygen and it's released. And it's carbon dioxide. Okay, so in stage 2, 2 carbon dioxides were released. And in stage 3, 2 for every turn of the cycle, but the cycle has to go around twice. So that'd be a total of 6 carbon dioxides is released from 1 glucose molecule. That's kind of easy to remember because look, the whole point of respiration is to break the carbon dioxide, break the glucose up. So basically what you're doing is taking the 6-carbon sugar glucose and breaking it up into 6 carbon dioxide molecules. So this is just reminding us that if aerobic respiration ended here, we'd only have 4 ATP molecules. Those are the useful energy molecules. You cannot use NADH and all that, and FADH too. You cannot use that in a cell directly for energy. You only can use ATP molecules if you need energy in a cell. So it would be a total waste to not be able to use NAD and FADH to it. And that's what would happen if you could not go into the 4th stage, the oxidative phosphorylation stage, OP stage. Okay, during the oxidative phosphorylation stage, which is stage 4, this is where the FAD and NADH are going to be cashed into ATP. Okay, so that's a very important step. You got lots of FADH and NADH having been charged up in the citric acid cycle, the Krebs cycle. So let's keep a running score. Stage number in aerobic respiration, the number of ATPs formed, charged up, the number of NADH charged up, the number of FADH2s that get charged up in each stage. So in the first stage, glycolysis, we only have 2 ATPs and 2 NADHs that are charged up. In the PVA oxidation stage, PO stage, second stage, we have 2 NADHs charged up. That's it. In the third stage, the Krebs cycle stage, we have 2 ATPs for 2 turns of the cycle. 2 turns of the cycle, we got 2 ATPs formed, 6 NADHs, and 2 FADHs. So, in the oxidative phosphorylation stage, stage 4, this is where all these NADs get charged up. So we got a total of 10 NADHs. 10 times 3 is 30, because each NADH is going to be worth 3 ATPs. So it gets like currency exchange. It gets cashed in. So that'd be 30. Also, in the fourth stage, FADH2 gets cashed in. Each FADH2 is worth 2 ATPs. So we got 2 there. So we got 2 times 2 ATPs is 4. So we count all the ATPs. 2 plus 2 is 4. Total. We didn't produce any. We don't produce. Those are already there from the stage 3 and stage 1. So we got 4. We got 30. So 30 plus 4 plus 4 comes out to be a total of 38 ATPs formed from the energy released, and 1 glucose was broken down by aerobic respiration. So we added all those ATPs. Now, if you did not have that last stage, you'd only have 4 ATPs. So that last stage is really important. Let's talk about that last stage. So that last stage involves an electron transport chain. Okay, we talked about this. The high energy electrons from FADH2 and NADH fall down this. These are electrons from these molecules. They fall down this electron transport chain in the fourth stage. And by the way, that's in the inner membrane. This occurs on the inner membrane, the cristae of the mitochondria. Okay, so as those electrons fall down, the ETC, they release energy, and that energy is used to charge ADP up into ATP. Okay, at the end of the chain is oxygen. At the very bottom of the chain, the final electron acceptor is oxygen. Now, if oxygen is not down there at the bottom, the electrons won't fall down the ETC, and you won't get that fourth stage occurring. So oxygen is really important at the bottom of the ETC. If we don't have oxygen, we die. We can't do anaerobic respiration for a sustained, for but a few, maybe a couple of minutes, and you'll die because you're not getting enough ATP to run all the activities our cells have to do to keep us alive. So right here, I did a clearer calculation of the total ATP back 2 slides back. Okay, let's talk about the electron transport chain a little bit. Okay, so this is the inner membrane of the mitochondria. That's where the stage 4 is, electron transport chain. Now, let's see. This is the matrix side. The matrix would be all that liquid inside the mitochondria. This would be the intermembrane space. That's the space between the inner and the outer membrane. Okay, so if we had mitochondria, here's all the squiggly stuff. Here's the inner membrane. That's the inner membrane. Here's the outer membrane. Here's the inner membrane space between the inner and the outer membrane, and here's the matrix. So here you see NADH and FADH2. These high energy molecules are going to be losing their hydrogen and their electrons. Some electrons, those high energy electrons, will be moved down this electron transport chain. You can see some of it. Okay, they don't show very well the electron transport chain, but they show some of it. So what's going to happen is those electrons and hydrogen is going to be released. The electrons, the high energy electrons, will fall down this electron transport chain. The energy release will pump the hydrogens into and concentrate them into the intermembrane space right here. So right here, we got a high concentration of H+, and because they're getting pumped from the energy released by the NADH and the FADH2, you'll get a low concentration of hydrogen ions here. So the energy release is being used to active transport the hydrogen over there. Now you might say, well, where's ATP come into here? How's that cashing in the energy? Seems like the energy is just being used to pump the hydrogen. What a waste. Well, here's this very important enzyme here called ATP synthase. In ATP synthase, well, those hydrogen ions will diffuse. That's not active transport. They will diffuse through this big enzyme. And when you diffuse, you release energy. And that energy is used to charge up ATP. So eventually, the energy is stored in the ATP molecules. So these two molecules right here, FADH2 and NADH, their energy is used to pump hydrogen ions, but then those hydrogen ions will move from a high concentration to a low concentration, and through a special channel protein, which is hitched to an enzyme and ATP synthase, which charges up the ADP to ATP. Oxygen, like I said, is a very final electron acceptor molecule at the end of the ETC, at the bottom of the ETC, where the electrons from NAD and FAD are being passed down in order to provide the energy to power the proton pumps, the H plus pumps. After that, oxygen picks up the electrons and two hydrogen atoms from the NADH and FADH2. It will form water molecules. And that's where, in the equation, C6H12O6 plus oxygen, because that's where the oxygen comes in the aerobic respiration equation to form H2O plus carbon dioxide. We already showed where the carbon dioxide formed. This is where the water is forming. So without oxygen, this whole thing can't make the ATP. And the oxygen will be used up and turned into water, which enters a mitochondrial, that remains in a mitochondrial matrix and eventually diffuses into the bloodstream. This process of using proton pumps to create a concentration gradient so that the protons from the high concentrated area can move through the ATP synthase channel protein and release energy that will energize ADP into ATP. This process also occurs in photosynthesis in the ETC during the light reactions. So this process that I just described is called chemiosmosis. Really has nothing to do with osmosis, but this is how most ATP is formed. So if you have an electron transport chain, you're harvesting ATP by the process of chemiosmosis with ATP synthase, allowing protons to passively transport through it, releasing energy that will be captured in ADP, turning it into ATP. The final electron acceptors in the ETC, that's oxygen. Remember, oxygen plus electrons from FADH2 and NADH plus hydrogen ions, protons, and the matrix forms H2O. So this O is from oxygen, so oxygen is the final acceptor. Okay, I went to pains to show you exactly why 38 were formed, the maximum 38 ATPs from one glucose molecule. Now, fermentation is called anaerobic respiration. You don't have electron transport chain. You don't use the inner membrane with the ETC of the mitochondria. You don't use oxygen. It makes a very small yield of ATP, not 38 per glucose, more like 2. So very inefficient. Okay, we use it when there's no oxygen available. Like I said, when we're sprinting, doing bursts of energy. The first step of fermentation is just like that of aerobic respiration, glycolysis, where you got the 6-carbon sugar being broken down into 2 PVAs. And remember, it was you get 2 ATPs and 2 NADHs from that energy released. Now, because there is no electron acceptor, no oxygen, in other words, at the end of the ETC, the NADH that's produced here cannot be cashed into ATP. And you're not going to be able to form more ATP here unless you have the uncharged, the unenergized NAD plus available. So what seems really wasteful, what has to be done is that NADH that was formed during glycolysis has to be used up. And what it does in our muscles or sprinting, it converts to NADH, the energy being used to turn PVA, those three carbon molecules that were produced by glycolysis, into 2 lactic acid molecules. Lactic acid has higher energy than PVA, and it's kind of wasteful that it's produced because you're wasting all that energy in the NADH, which is worth 3 ATPs each. So you're just throwing 6 ATPs of energy, that energy into this molecule of lactate. So in our body, eventually lactic acid will be broken down and go into the Krebs cycle. But in organisms like in other more primitive organisms, that lactate just is wasted. Okay, so the whole point of this wasteful step here is return the NAD plus there, because that has to be changed into NADH first before ADP turns into ATP. So if you didn't have this second wasteful step where NAD plus is regenerated, you couldn't go back to glycolysis and you'd have 0 energy then. Okay, there's 2 types of anaerobic respiration, which we call fermentation. There's lactic acid fermentation, that's what our muscles do. And there's ethanol fermentation. This is what yeast does, primitive organisms like yeast. This is ethanol fermentation or alcohol fermentation. Okay, so like yeast does. Okay, so it's the first step is the same. Glycolysis, same. Now, what happens, instead of making lactic acid, the pyruvate is eventually turned into ethanol, which is drinking alcohol. Okay, carbon dioxide, ethanol. Carbon dioxide is also produced as a waste product. So the whole point of this is to get NADH back to NAD plus so that you can recycle it and do that step up here in order that you can make ATP. The net result is still only 2 ATP per glucose molecule, just like it was for lactic acid fermentation. Okay, but it produces alcohol. That's how we make beer and liquors. We use yeast and sugar from juice. It makes wine, beer, and hard liquor. So carbon dioxide is also useful. We use yeast for making bread. Okay, the dough rises because the carbon dioxide builds up and puffs up the dough. Okay, the alcohol is in that dough too, and it cooks off. The alcohol evaporates while it cooks. That's why bread smells so good when it's baking. You smell the alcohol coming off. So you get the same net gain of ATPs, whether you're using lactic acid or ethanol fermentation. You only get 2 ATPs per one glucose molecule compared to aerobic, where you get 38 ATPs per glucose molecule. Big difference. Here's a little details. Some scientists disagree that it's 38 ATPs. They think it's 36, 37 for aerobic because you have to move the NADH through a membrane, and that's the active transport process for the NAD that comes off of steps 1 and 2. So that takes some energy to do that. But let's keep with the maximum is 38. So cellular aerobic respiration is quite efficient, 55% efficient. Again, because the second law of thermodynamics, 45% is lost as heat when this happens in the mitochondria, the 4 stages. In fermentation, only 3% energy in glucose is used. The remaining 97% of energy is lost as heat. Very inefficient is fermentation. But if you're a simple organism, no big deal. You can handle it. Aerobic respiration is very efficient. You might say, no, it's 55%. Well, if you're running a factory and you have 55% efficiency, you'd be making a lot of money. Okay, you can read this. Bacteria that are aerobic don't have mitochondria, so they can't do aerobic respiration, but they can do fermentation, and that's what that's why they use making cheese and yogurt. They add to the flavor of the cheese, and they also digest the milk and to produce the cheese and the yogurt, but they do it by either lactic acid fermentation. That's usually what they use. Okay, yeast will do ethanol fermentation, because when you put yeast in the dough or the water in juice, what will happen is that there won't be oxygen in that, and it will act like a bacteria without a mitochondria, even though yeast does have a mitochondria. So it will do ethanol fermentation, release carbon dioxide, and produce alcohol. In our muscles, we do lactic acid fermentation if we don't get oxygen. And lactic acid is what causes that soreness after you work out hard without breathing enough oxygen in. Okay, so glycolysis followed by lactic acid fermentation. Okay, so we're talking about lactic acid fermentation. The final products are 2 ATPs and lactic acid. That's it. No carbon dioxide. That's ethanol fermentation. NADH, well, it did produce NADH, but it flipped it back to NAD. So it did not have a net gain of any NADH. So it'd be 1 in 5. Bread rises due to the production of, okay, that'd be ethanol fermentation, but the ethanol isn't what's causing the bread to rise. It's the carbon dioxide byproduct that's causing it to rise.